Welcome back. In the last lecture, I talked about genetic capacity in terms of protein-coding genes. And these protein-coding genes then determine the phenotype of an organism. Now I want to extend this by looking at the whole genetic capacity of an organism in terms of the entire sequence of the nucleotides that are there in the genome. And the genome is the entire complement of genes that make up an organism. My story is the history of genome sequencing, and it begins in the early 1900s when genetic mutations were first studied in the model organism of the fruit fly. That's the supermodel of genetics. Scientists found there were a number of things that would increase the rate of mutations. You'll recall that mutations can happen spontaneously when DNA replication makes errors. Mutations can be increased if something damages DNA. And you'll recall in our last lecture that I defined DNA damage as mutation, a base pair change. One of these mutagens, things that cause mutations, was ionizing radiation. And we really found out this the hard way. Miners working in uranium mines got cancer because the uranium caused genetic damage, radiation leaking from it, and the genetic damage led to cancer. People making luminous watches in the early decades of the 20th century. To glow in the dark, you need some energy source, and the energy source they used was radium. And these people would use very fine brushes, camel's hair brushes, to paint the dials, and they dabbed them with their tongue to get the brush together to paint the dial. And of course, they got oral cancers. By the 1930s, this type of mutagenesis due to radiation had been studied enough that we could draw a graph, and the graph would have radiation on one axis and damage of genes on the other axis, and the more radiation you got, the more damage of genes you got. In the closing days of World War II, the United States exploded atomic bombs on the Japanese cities of Hiroshima and Nagasaki. Hundreds of thousands of people were killed. Many more were exposed to radiation as what is called fallout, particles of radioactivity that then gently came down to the ground. These survivors and their descendants have been intensively studied ever since that time for increases in mutations of somatic cells. Remember, the vast majority of cells in our body are somatic cells that are not sex cells. And here, genetic damage can lead to cancer. And germline cells, so that we're looking at the offspring of people who were exposed, and now their great-great-grandchildren in some cases have been born. And you're looking for genetic abnormalities in the next generation. By the late 1970s, methods had been developed to get the sequence of DNA molecules. But you know, DNA molecules are very large, and the only way we can sequence them now with current technology is 800 base pairs or so at a time. So we can sequence fragments of DNA, 800 base pairs at a time. And you can do that even by machine now. You used to do it manually, you can do it by machine. Until that time, the scientists who were studying genetic damage in the Japanese survivors and their descendants looked at genetic damage by inferring genetic damage from phenotype. People got more cancers, for example, thyroid cancers and leukemias, who were survivors of atomic bomb radiation. And so you surmise then that there's genetic damage in DNA. And some of their offspring had some genetic damage as well, and you looked at it through the phenotype. Well, now that we could sequence DNA, we could look at the genes directly. We could look at the base pairs that had changed due to damage. In 1984, a Nobel laureate, Renato Del Becco, who had won the Nobel Prize for studying cancer viruses, made the daring suggestion that we should sequence the entire human genome, because if we did that, we could compare the entire human genome, all the DNA, all 23 chromosome pairs of a human, had all the nucleotides. We could compare the people who were not exposed to those who were exposed, and we could get a very good handle on genetic damage. It's not surprising then that the first sponsor of what came to be known as the Human Genome Project was the U.S. Department of Energy. U.S. Department of Energy oversaw the radiation damage project. Since then, it's been farmed out to other organizations. The human genome sequence was determined in two ways. There was a huge challenge here in genome sequencing. The problem was the following. Chromosomes are extremely long. Typical human chromosome, on the average, 100 million base pairs of DNA. These machines can sequence, get the nucleotide sequence from start to finish, 800 base pairs at a time. So what do we got to do? We'll cut the 100 million base pairs into 800 base pair fragments. How many is that? That's your assignment. Please email me or the teaching company with the number of fragments. It's a lot. Now we determine the sequence of each one of those fragments, and then of course you just fit them together. Oh yeah? How are you going to fit them together? How do you know which fragment is which? Because we're not cutting it end on. We're cutting it randomly. So we're taking the DNA and we're hitting it with a restriction, with some sort of enzyme, like you'll see later on in the course, restriction enzymes that will cut DNA at certain places, or we can just physically cut it. It doesn't matter. We're not cutting it end on. We're just cutting it whole piece into 800 base pair fragments. It's kind of like taking the printed guide for this teaching company course, cutting out each word, throwing them all up in the air, putting them down on the floor, and then lining them up to make the sentences I have written. Good luck. I think it's going to be very hard to do. There were two methods to get these to get some sort of signposts by which we could say this particular 800 base pair fragments on the left end, this one's in the middle, this is on the right end. Okay, that's the simplest way to look at it. The first method was called hierarchical sequencing, and the second method was called shotgun sequencing. The hierarchical sequencing was done by the government-sponsored Human Genome Project. And this was not only sponsored by the US government, it was sponsored by a consortium of governments from around the world. Thousands of scientists worked on this effort. It was led by a physician scientist, Francis Collins, and still is led by him. What they decided to do was identify short sequences of DNA, a couple of base pairs long, that were at certain landmarks all the way down the chromosome. So if that short sequence is on the left end of the chromosome in the first 800 base pair fragment, and then you randomly sequence fragments and you see that short sequence, you'll say, oh, that's the one on the left end. These were the signposts they used. It's kind of like saying, well, supposing we were doing the word problem that I just had. You've got all the words for this manual, and they're all on the floor, and we need a signpost to say, how about lecture 2? Where is lecture 2? Well, that's going to be about the middle of our the lecture 2. The word Mendel appears in lecture 2. So you look on the floor and say, Mendel, oh, that's over here on the left, near the start of his lecture series. That's going to be in the start of the manual because indeed that's where Mendel appears, and we don't use Mendel's name anywhere else. Well, I just used it, so now we're in trouble because Mendel appears several places. Okay? So we had these short sequences. It took 10 years for scientists to look at all the human chromosomes and get these landmark sequences all along the chromosome. And there were thousands of them all along the chromosome. Then what they did was cut the chromosome into 800 base pair fragments, sequence all the fragments to 800 base pairs, look for the landmarks, the little sequences, and then arrange them along the chromosome, each chromosome. Took about 12 to 15 years, depending on how you define it, to do this human genome sequence. The other way of doing it is called shotgun sequencing. Shotgun sequencing was done by private industry, led by a scientist named Craig Venter. And what they did was the same thing using landmarks, only they had a computer find the landmarks. And so what they did was take the chromosome, bust it into 800 base pair fragments, sequence them all, feed the sequences into a computer, and have the computer line it up for you. This took amazingly sophisticated computer analysis. A whole new field was invented for this shotgun sequencing effort, and that field is called bioinformatics. It's a major field of computation now. The computer would analyze a sequence saying, oh, that's on the left-hand end. Mendel is there, for example. And that's going to be in the left-hand end of the chromosome, and this is on the right-hand end of the chromosome. This was much more rapid. It took only a year to sequence the human genome using shotgun sequencing, as opposed to 15 years doing it the painstaking way. Having the computer do it was a lot faster. But when the people started the hierarchical sequencing, the Francis Collins-led Human Genome Project, originally the computational power wasn't there. The computer programs just weren't there to analyze DNA at that level. Now the computer programs are there, and so shotgun sequencing is much more common. What information can we get from genome sequencing, from getting all this A, T, G, C, 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 etc.? There are several types of information we can get. The first type are called coding sequences, open reading frames. A computer can scan along a gene sequence, not a chromosome sequence, along this long chromosome sequence, and the computer scans along and says, oh, look, that's a promoter. I've seen that before. Oh, look, that's the codons coding for amino acid. Codons are those 3 base code words in messenger RNA. So those are the code words or codons along the DNA that code for proteins. And those are different than random DNA sequence because they've got the promoter there. The promoter is kind of a signpost saying gene starts here. It's kind of like a capital letter starting a sentence or indentation starting a paragraph. We have that language. The second thing, of course, you can see, once you've got the code words there and the DNA, you can look up the genetic code and say, well, here's the protein that thing makes. So you can have that information. Related to that is something called, technically called annotation, where you can find the function of that protein. So now we have a genome sequence, we have the gene sequence, now we have the protein function that gene codes for. And of course, you can get gene control information from the promoter. In context of the Human Genome Project, a number of non-human genomes have been sequenced. And most of these are on the model organisms scientists study to represent other organisms in its class in nature. First, the model organism was sequenced, and then other organisms afterwards. There is a huge amount of information we can get from this. We can do what is called comparative genomics. When a gene is sequenced, or an entire genome is sequenced, the information, of course, is done on computer, and it goes to a big database, and there are a number of these in the sky. It's not really in the sky, it's at computers, usually in Washington or some other place. In Europe, there are a number of places that have repository computers for storing genetic information, and the amount of information is awesome. We have way more information than we have analysis now. Analysis is behind the information. It's easy to get a sequence, much easier than it is to get the information from it. So supposing we have the sequence of a human gene. We don't know what that protein does. We have the sequence of the protein, and it's hard to predict in some cases, just from the amino acid sequence of a protein, how it's going to fold and what it does. Well, we dial up the database and we say, is there a similar protein to this somewhere else in nature? And you might say, well, there is a protein. It's a receptor for a hormone or something in the fruit fly. If that's true, then maybe it's the same hormone receptor in the human. Or it might have a different function, and we're looking at evolutionary change. Non-genome sequences began, non-human genome sequences began with the bacterium. The first genome to be sequenced is in the context of molecular biology a long time ago, 1995. This was a bacterium that causes meningitis. The bacterium is called Haemophilus influenzae. And just let me give you the data because you'll see how complex things got. It had 1,830,137 base pairs, 1,743 protein-coding genes. Half the genes until that time were unknown. So we immediately doubled the amount of knowledge about this thing. This thing causes a meningitis, so it was very interesting. When that was published, it was a sensation in biological thinking. It opened up the whole door of genome sequencing. I had never thought in my career as a scientist I would ever see this done. Many new genes have been discovered in these prokaryotic genomes. For example, genes for virulence or disease causing the typhus causing bacterium. The gene for attaching to human tissues in the bacterium that causes tuberculosis. So if we have that gene and we know it's protein, we could make a vaccine. Genes for attaching to plants in bacteria that fix nitrogen. You remember I told you that nitrogen gas in the air cannot be converted into amino groups except by some organisms, most notably bacteria. Now we know how they do it, and we know the genes involved through the genome sequence there. Well, one step up from bacterium, and many bacterial genomes have been sequenced now, is the model eukaryotic cell yeast. Yeast genome has 12 million base pairs. We only had 1.5 million in bacteria. 12 million base pairs, 6,000 genes. It's got all the genes for bacteria, for basic metabolism, for building up and breaking down substances, but it's also got genes for being a eukaryote, such as building compartments in the cell, like the nucleus, and targeting proteins to those compartments. The model complex organism is a worm. I've mentioned this before. This particular worm has only 1,000 cells, but it's an amazing worm. It's transparent, it's very small, a couple of millimeters in size. It only has 1,000 cells, but in those 1,000 cells is a reproductive system, a basic nervous system, a little bit of a digestive system. It's got organ systems. It's complex. All those 1,000 cells form from a single cell. You can watch it happen over 3 days. Amazing amount of information we can get from this particular organism. This organism has 97 million base pairs in its genome. Now we're getting a little bigger. And 19,000 protein coding genes. And a lot of these genes are doing similar things to making a complex body with organ systems like we have. A model organism which led to genome sequencing of rice, a very important plant genome. The rice genome, well, we're making a real step up now, 430 million base pairs. All these sequences have come out in the last 5 or 6 years. And 35,000 genes. So we've learned a lot about the genes of rice plants, and we'll talk about that when I talk about agricultural genetics in lectures 23 and 24. The human genome, of course, has been sequenced. The two groups, the publicly funded Human Genome Project and the privately funded group, announced their drafts of the genome at the same time in the year 2000 to great fanfare. It was a White House press conference with the president, etc. And Craig Venter and Francis Collins were there with President Clinton. The final draft came out in the winter of 2003. The human genome has several characteristics. One, of the 3.2 billion base pairs in the human genome, less than 2% code for proteins. 98% of the genome is not coding for proteins. You're saying, what's going on here? We'll talk about that in a minute. There are 24,000 genes in a human. I just told you rice has 35,000 genes. Humans have 24,000 genes. I consider that a major affront. I think I'm more complicated than a rice plant. How come I have so few genes? We'll talk about that problem in a moment. The average gene size in protein coding is 27,000 base pairs, including the promoter. Over half of the human genome has short, repeated sequences that don't code for proteins. 99%, give or take, of the human genome is identical in all people. 1% is different. Look around the room where you are. Look in your car at the other people driving. Yes, you are different in your genes, as well as the environment that influences that. Well, 1% of the human genome is a lot of base pairs that's different. The functions of many, or well, not some of these, good chunk of these 24,000 genes coding for proteins in humans are not yet known, so we're trying to find out what they do. And other organisms have sequences very similar to us. The worm I just talked about. Remember, the worm has 19,000 genes. We have 24,000 genes. There are a bunch of genes in the worm that are very similar to genes in us. Humbling again. Genome sequencing is getting faster and cheaper. In 2007, the genomes of James Watson, the discoverer of the DNA structure, and Craig Venter, the guy who sequenced the human genome by the private method, were made public. It took those genomes 2 months each to get sequenced, just 2 months, not a year now, and it costs about $2 million. The price is going down and down and down. Within a year or two, as I speak, it may be about $10,000 to sequence the genome. And the objective is within the decade of the by 2010 to 2015, there may be a $1,000 genome sequence effort. It's possible. Why do it? Screening for possible diseases. We'll talk a lot about this. Research, finding out what humans are, what they're made of. Origins, where did we come from? We'll talk a lot about that when I talk about evolution. There's a problem of too few human genes. I am insulted that I have fewer genes than a rice plant. Of course, as I tell my students, of course, tenured professors have many more genes because of our complex brains, and once again, they put down their pens and say, he's got to be kidding. No, we've all got the 24,000. There are three explanations, possible explanations for this anomaly of so few genes in humans, yet such a great amount of complexity. First, something called alternate RNA splicing. Genes are interrupted in their information sequence by non-coding sequences. These intervening sequences, called introns, are kind of nonsense. Now you might say, what are they doing there? There are explanations, but I don't want to get into that right now, just to tell you that they're there in the gene. So for example, I could say, this course is good. That's information, correct? But if this was the analogy of a messenger RNA at the ribosome, it would say, this course is good. That's a meaningful statement. In the DNA that codes for this course is good might be all these intervening words. So this teaching company, hair follicle, left toe, course, president, vice president, secretary of the Senate, is dirt, air, water, good. Now we have to get rid of all those nonsense words to make sense out of this. And that is done in the nucleus. But it has this cutting and splicing to get the word this, course, is, good, get them together, happens in the nucleus, happens at signals. But we could, theoretically, put those words together differently, couldn't we? We could just put this course is, leave out the word good. Or we could put this is good. That's called alternate splicing. And it happens to make proteins. In fact, every protein coding gene has so many of these internal introns or intervening sequences that the coding regions can be mixed and matched together. Each human gene gets translated into about 5 proteins. So we don't just have 24,000 protein coding genes. Those 24,000 can make about 120,000 different proteins. Second way that we can deal with this problem of 24,000 is called post-translational modifications. After a protein is made and completed, it folds into its three-dimensional shape, but other things happen to it. Sugar coating can happen. So it can't just be a plain donut. It could be a sugar-coated donut, which tastes quite different than a plain donut. So sugars can be put onto proteins and make them have different functions. So we can have a whole set of genes that add sugars to various proteins. So a protein can add 3 sugars in one place and 5 sugars in another place and have a different function. Proteins with sugars on them very often act as flags on the outside of a cell that causes the cell to bind to and recognize to other cells. It's important in cancer. So these post-translational modifications add to the diversity of proteins that can be made. Just making a protein is not enough. You could add things to it to change its structure and function. Third, there is a chunk of the 98% of the human genome that actually makes RNAs that don't get out of the nucleus, that don't end up getting translated into protein, but that stay in the nucleus. These are very small RNAs called microRNAs, and they're very recently discovered. In fact, they're not even in most textbooks. In the eukaryotic, in the human genome, there are many of these small RNAs that seem to be made by the genome, and humans make a lot of these. And these RNAs may be involved in gene regulation, in turning a gene on or off, in having that gene make a protein or not make a protein, make a messenger RNA or not make a messenger RNA. We're not quite sure what the microRNAs are doing, but humans have a lot of them, and they are obviously an informational part of the genome. You know, scientists used to say that the 98% of the human genome that doesn't code for proteins is junk. Well, looks like this junk has a valuable function. The junkyard does have some gems there, and the gems are these microRNAs hanging around the nucleus doing something we think is pretty important in controlling genes. So that's an added complexity. So we may get away with 24,000 genes because we do so much with them, and we can do many, many more things than we thought. The Human Genome Project has led to a new frontier. And that frontier became realized when the most simple prokaryote was sequenced. The genome of Mycoplasma genitalium. This is a small prokaryote that, as its name implies, genitalium, lives in the genital tract. This genome is the smallest in nature. It has 482 genes, not 24,000, 482 genes. Using technologies of manipulating DNA, some of which I will describe in future lectures, scientists have been able to inactivate these genes one by one and ask the following question: does the cell survive? So you've got this cell with 482 genes, and you take gene 1 and you knock it out. You ask the question, okay, are you still alive? And if the cell is alive, you say, you know, it really doesn't need that gene to survive. If the cell dies, it says, oh, it's essential for survival. In that way, scientists have knocked out 100 genes, and the cell still survives. So there are 100 genes that are dispensable, and 382 genes that are indispensable for life. Wow, we are now down to a genome of 382 genes which are essential for life. You need all 382 of them, and you don't need much more to be alive as a very small bacterium living in the genital tract, but still alive. What are these genes? These are genes that code for proteins involved in basic cell structures like the nucleus or in a eukaryote or the ribosome, and their functions of cells like enzymes for energy or making RNAs and making proteins, etc. Obviously things that all cells have to do, make the structures and functions of every cell. Craig Venter and his colleagues in a private company are trying to make each gene of these 382 in the chemistry lab. As I'll describe in a later lecture, it's possible to do that. You can go in the chem lab and make genes. You can do that by just taking it, say, the gene has the following sequence, A, T, G, C. You go in the lab and you say to the machine, take an A, add it to a T, add it to a G, add it to a C. That's just chemistry. It's Tinker Toy stuff, but it's chemistry. So you can make each one of these genes. Then they want to put all these genes together, 382 of them, in a genome, artificial genome, put them in an empty cell and create life. Is this just a stunt? No, because if you can do that, you could take any gene you want and put it into this prokaryotic cell. For example, you could have the prokaryotic cell make ethanol for you if you put the genes for the biochemical pathway there. You could have it degrade oil in what is called bioremediation for oil slicks. You could have it make plastics for you. There are many things you could do. You could also, of course, have it make ricin, which is a toxin. And so there are very scary biological warfare aspects of this brand new field, which is called synthetic biology, making artificial chromosomes by artificially making DNA because we know the genome sequences, and then putting it into an empty cell and having that cell do the things that you want. Knowledge of DNA sequencing, as I've just described, and knowledge of DNA in general, has allowed scientists to manipulate it, and I've just described one of those manipulations. In the next lecture, I'll step back and talk about one of the major manipulations of DNA, which is called recombinant DNA. Thank you.